Hi friends, welcome back to my channel Only Bio. Presenting up the series of the challenging MCQs from Group A 66 to 70. The first question relates with the chapter circulation and the question is: which of the following causes inhibition of the rate of impulse produced by the SA node when the heart is treated with it? And your options are A. Acetylcholine, B. Adrenaline, C. Sympathin, and D. None of these. And the correct answer for this question is acetylcholine. Friends, I hope you remember something about the SA node, which is very rightly called as the pacemaker of the body. What is SA node exactly? It's a cluster of myocytes, right? And it is present at the junction of the crista terminalis in the upper wall of the right auricle or right atrium जैसे आप कहते हो, right? And the opening of the superior vena cava. So in a healthy heart, SA node continuously produces the action potential which sets the rhythm of the heart, and this is the reason why it is called as the heart's natural pacemaker. Right? Now this SA node may dysfunction at times, and this can result maybe from ischemia or necrosis to the pacemaker cells due to decrease in the arterial blood supply or something like that. Now, this SA node, when stimulated by the parasympathetic nervous system, it slows down the rate of the action potential production, which thereby decreases the heart rate. Whereas, if it is stimulated by the sympathetic nerves, this increases the heartbeat. ये आपने शायद पढ़ा होगा ना. यहाँ पर जो ऑप्शंस दिए हुए उसमें से है जो एसिटाइल कोलिन, दैट्स द राइट आंसर. See, when the vagus nerve, you know that's the 10th cranial nerve and very important cranial nerve called as a wandering nerve, right? इसके stimulation पर it releases acetylcholine which reduces the heart rate till it reaches the normal sinus rhythm. It is believed that it does so by increasing the potassium conductance of the pacemaker cells in the SA node. Now the option B, adrenaline. You might be remembering that adrenaline is released up as a response to the fear or stress or excitement or shock kind of a factor. It's a hormone which has a direct effect on the SA node and it increases the heart rate. That is just to prepare the body for any physical activity. The third option that is given is C, that is sympathin. Now friends, please remember sympathin refers to the mixture secreted by the sympathetic nervous system, mixture of adrenaline as well as noradrenaline, and both of this collectively we call it as sympathin because it is released by the sympathetic nervous system, and both of these have an impact on the SA node, that is it increases the heart rate. I take you to the next question. The next question relates with the chapter human respiration, and the question is: an increase in the carbon dioxide concentration shifts the shape of the oxyhaemoglobin dissociation curve to the right. This phenomena is known as dash, and the options given are A. Bohr's effect, B. Hamburger's effect, C. Newton's effect, D. None of these. And the correct answer for this question is A. That is Bohr's effect. Friends, what exactly is Bohr's effect? You can look on the screen, it's written Bohr's effect was being given by a scientist Christian Bohr, who stated that the oxygen binding affinity of hemoglobin is inversely related to the concentration of carbon dioxide and H plus ion concentration. Now, what exactly do we mean by this? Let's understand this a bit better. Now, Bohr's effect जो है, okay, I mean in simple words, you can say it is the hemoglobin's oxygen binding affinity which we are talking about. Okay, so the Bohr's effect refers to the shift in the oxygen dissociation curve that is caused by the changes in the concentration of the carbon dioxide or the pH of the environment. Now, what exactly happens? Dekho, when carbon dioxide reacts with water to form carbonic acid, an increase in carbon dioxide results in the decrease in the blood pH. It becomes what? It becomes more acidic, and this results in hemoglobin proteins releasing their oxygen. Okay, so on the contrary, a decrease in the carbon dioxide will provoke what? An increase in pH, and this in turn will result in hemoglobin picking up more oxygen. You can look on the screen wherein you can easily understand what exactly is the Bohr's effect all about. Here you can see, I'll take up the pointer. You can see that this is a so-called graphical representation, and this line represents the oxygen hemoglobin dissociation curve. This deflection here is towards the right side. अगर ये line यहाँ झुकी होती, if this line would have been deflecting here, I would have called it as the left shift. But here we can clearly see that this particular part of the line is mainly shifting to the right side. Hence it is called as the right shift. And this can happen because of decreased affinity for oxygen. That is when the pH goes down or if the temperature is higher. And on the contrary, the left shift is because of the hemoglobin's affinity for oxygen being changed. Now what happens is there's a decrease in the partial pressure of the carbon dioxide. Concentration of H plus ions is also affected. Now here in the options that have been given, we also have come across something called as Hamburger's phenomena. Right now, Hamburger's phenomena, yeah, Hamburger's effect is nothing but it is more commonly called as a chloride shift, which is a process which occurs in our cardiovascular system and refers to the exchange of bicarbonate and chloride ions across the membrane of the RBC. You can look on the screen. This is a RBC that is given here, that has been enlarged. So this is a RBC. Carbon dioxide from the tissue enters inside the RBC. It reacts with water in presence of carbonic anhydrase enzyme to form carbonic acid plus H plus. Now what happens is this H plus, it binds to hemoglobin to form HHBO2 and oxygen is released out, and the carbon dioxide also may react with hemoglobin to form HbCO2 plus O2, and the O2 is released out in the plasma. Now what happens is the HCO3 that is formed is exchanged outside. Now here you have to remember that plasma has a good concentration of NaCl where it can undergo dissociation to form Na plus and Cl minus. So when bicarbonate ions move out, okay, when here you can see the bicarbonate ion moving out, at that time the chloride ion moves in. That means there is a mutual exchange here, a shift here, and this is the reason why it is called as the chloride shift mechanism. So as the concentration of bicarbonate increases in the RBCs, it diffuses into the plasma along with the concentration gradient in exchange for the chloride ions, so that it maintains the electrical neutrality. Okay, this phenomena is called as the chloride shift, which helps to regenerate the bicarbonate ions. And there was also another option called as the Newton's effect. Let me tell you that has got nothing to do here. Okay, Newton's effect, of course, it will relate up with your, what you may say, physics. Now, the next question relates here with the chapter that is sexual reproduction in angiosperms, and the question is, the reason why a flower is considered as a modified shoot is that, and the options are A. Flowers group together to form an inflorescence. B. In some flowers, thalamus becomes elongated and shows its distinct nodes and internodes. C. Epicalyx is present in some flowers. And D. Shoots of some plants become non-green in color. And the correct option for this question, it's quite easy, and the answer is B. That is, in some flowers, thalamus becomes elongated and shows its distinct nodes and internodes. You can look at this as a typical flower structure, and the thalamus, which is also at times called as the receptacle. At times it is asked. Next question relates with the chapter genetics, and the question is, you can even relate this up with the chapter evolution. Let me tell you before I start. The question is, for the determination of variations, whether they are genetically controlled in a population, the most appropriate procedure will be dash, and your options are counting of chromosomes, B. examination of DNA, C. measure the variations and see if they are continuous or discontinuous, and D. cross individuals of both extremes and see if the offsprings and parents show the same range of variations. And the correct answer for this question is D. Friends, you know genetic variation is an important factor in evolution, and this allows the natural selection to increase or decrease the frequency of alleles in a particularly existing population. Genetic variation may be caused by mutation, so which can bring about entirely new alleles in the population, or it may be because of random mating, random fertilization, recombination between homologous chromosomes during meiosis. Now, genetic variation is advantageous to a population because it enables some individuals to adapt to the environment while maintaining the survival of the population. I hope you have understood this. I take you to the next question. The next question relates with the chapter excretion and osmoregulation, and the question is, the correct sequence in the formation of urea by mammalian liver through Krebs cycle and Henseleit cycle is, and the options are A. citrulline resulting to formation of aspartate resulting to arginine to urea, B. ornithine forming citrulline forming arginine forming urea, C. citrulline forming ornithine forming arginine forming urea, and D. arginine resulting in the formation of citrulline to ornithine to urea. And the correct answer for this question is B. That is ornithine resulting in the formation of citrulline, then arginine, and then urea. Please look on the screen here. See, the urea cycle, which is also, you might have heard about it as an ornithine cycle. Okay, it is a set of biochemical reactions that produces urea from ammonia. And this cycle occurs in ureotelic organisms. Now, you know, ureotelic organisms are those which excrete out the nitrogenous waste products in the form of urea. So, the urea cycle, it converts a highly toxic ammonia to urea for the excretion purpose. And this cycle was first metabolic cycle to be discovered by Hans Kreb. That means even before the discovery of the TCA cycle, that is the tricarboxylic acid cycle that we talk about in respiration. And this cycle was later on studied by many people. Now here you can clearly see on the screen that arginine forms ornithine here. Of course, it's a kind of a, what you can say, cyclic thing. Let us focus here on the mitochondrial matrix, the citrulline forming arginosuccinate, forming arginine and forming ornithine. So this is the last step jahan urea banta hai. So the cycle ideally starting up here. Okay, I hope you understood this. And friends, please share the link, like the video if you have really liked it, subscribe if you haven't, and hit the bell icon if you haven't. Thank you.